Thank you for joining us for Psomagen's introduction to 10x Genomics On-Chip-Multiplexing, also called OCM, and its utility in CRISPR screening. Before we dive into on-chip multiplexing, which works with both 3' and 5' GEMX technology, I thought it would be good to revisit how a gel emulsion works. And we're doing this with the 3' technology. It begins with 10x barcoded gel beads. These are the very expensive part of the assay here. They're fed into a chip along with individual cells, single cells, as well as a gel emulsion oil, with the goal being to form an emulsion droplet that contains both a single cell as well as one 10x gel bead. After that occurs, the cells are lysed, reverse transcription is conducted, and every single transcript from that single cell gets the same unique 10x barcode. After this step, the emulsions are cleaned up. There are many wash steps where there are significant losses, and then all the cDNA is pulled together for library prep and sequencing. After sequencing, due to that unique 10x barcode in UMI, all of the individual transcripts can be linked back to that original starting cell from which they came, and multiplets can be demultiplexed. Now, one of the limitations of standard 10x 5' or 3' single cell workflows is that you really need to start with at least 100,000 cells or nuclei, with the goal of recovering 20,000 cells or nuclei and loading that into the chip. And when they're all collected together, you have a total of 80 to 100,000 cells left. With on-chip multiplexing, you have the ability to start with a much lower cell number and pull as few as 5,000 cells each. And that's where the utility really comes in, both for working with things that have low sample types, like limited regions of mouse brain or organoids, or even CRISPR screening. Now, CRISPR-Cas9 screening is the one we're specifically going to talk about, and this is an enzyme that will make a targeted double-stranded break within genomic DNA. And each of the guide RNAs for Cas9 contain a highly conserved sequence. And when 10x designed their 5' single cell assay, they were very smart, and they designed it so that they could target that specific sequence, which is indicated by the little L-shaped with two double-ended beads between nexus and hairpin in the diagram. This is wise because then you can get your guide RNA library. You also, of course, always get your gene expression library to verify the findings of your CRISPR modifications. And then as a bonus, you can even get that VDJ library where you get the cell surface protein, and look at that with feature barcoding. Now, this is in contrast to the 3' assay, wherein that conserved sequence is not found within the default 3' library. You would have to modify the entire guide RNA library with either capture sequence 1 or capture sequence 2. That does not mean that 5' and 3' aren't suitable for on-chip multiplexing. They both are. So again, you can use those lower cell numbers to combine together, and you can still take advantage of that great 10x single cell sequencing workflow if you work with organoids. The benefit here for CRISPR screening, at least Cas9 library based, is that you have that ease of use directly out of the box with the 5' libraries. Please contact inquiry@psomagen.com if you have any additional questions or would like to try this out. We run this all the time.